Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamagni. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. And with me today are my colleague at NCCAT, Ed Eng. Joining from the Stanford SLAC Cryo-EM Center, S2C2, is Michael Schmidt. And from the Pacific Northwest Cryo-EM Center, PNCC, is Jeanette Myers. Our speaker today is Radhika Malik, who I'll introduce properly after my colleagues from each of the centers give a quick update on the resources we make available at no cost to the research community. This webinar is a regular event. We host the last Thursday of the month to highlight methods and tips and tricks that groups are using to collect and interpret the data they can obtain through use of National Cryo-EM Center resources. So you can save the date now for our next talk on March 30th. Details are still TBD, but look out for announcements for our March 30th talk. One purpose of this webinar is to help researchers across the US learn about the centers and how they can make use of them. Another way we do that is by attending conferences. So members of the staff from our centers, including Mike and Ed here, were just at the Biophysical Society meeting. Maybe some of you saw them there, or maybe some of your colleagues are currently on an airplane right now coming back from San Diego and missing this talk. And if that's the case, we do have our recordings available on our YouTube page or the events page at cryoemcenters.org. We've also been incorporating some informal roundtables into our schedule, and we're going to be doing that more over the course of this year. So if there are particular topics on any aspect of the single particle cryo-EM workflow you're interested in, please let us know. You can send us chats during this talk, or you can email us. You can find our emails on our websites, but let us know what you're interested in hearing if you'd like to take part in some of those more informal conversations. For final Zoom logistics, I'll be moderating questions at the end of the talk, but you can send them anytime using the Q&A box. So please use that Q&A and not the chat to send questions. And with that, we can go ahead and get started. So I'm going to turn over to Jeanette to give us an update from PNCC. Oh, and Jeanette, go ahead.
Yeah, I just realized I was muted. Yeah, here we go. I'm Jeanette, a microscopist at the Pacific Northwest Cryo-EM Center, or PNCC for short. Just a quick intro to what we offer. We have 2 types of proposals. One of them is our screening access proposal, which would let you come on site with us for up to 5 days of sample preparation and screening with staff assistants. And this is aimed at people who are from low resource institutions who might not otherwise be able to freeze grids, etc. The other proposal type is our general access, which will give you up to 480 hours of microscope time a year for 2 years. And that could be used for either screening or data collection, depending on what you need. And approved general access proposals are also eligible for training workshops, which will cover topics from sample preparation through data processing. And approved proposals are also assigned a scientific point of contact, or SPOC, like myself, who can help you with questions about sample optimization, data collection strategies, etc. And just as far as our equipment goes, we have 1 Icahn, 4 Cryos. All of them have K3 cameras. We have a Falcon 4i available if you're interested in using that. 3 out of our 4 Cryos have energy filters. And for sample prep, we have a Vitrobot and a Leica G2. And if you're interested in applying, our deadline is the 1st of every month, and I would love to hear from some more of you guys. And that's all from me. Bye-bye.
Thank you, Jeanette. Now I will turn over to Mike.
Hi, I'm Mike Schmidt, and I'm one of the co-directors of S2C2, the Stanford SLAC Cryo-EM Center. We have 3 Krios microscopes and one Arctica, soon to be 4 and 2, respectively, on screening scopes versus data collection microscopes. Our training, our screening, and our high-resolution data collection pipelines are all working and all deliver high-resolution results to people, no matter what what pathway they're using. We have trainees who are getting 1.9 angstrom resolution maps. So training has been very successful. Screening has been also very successful. All of our programs are running full swing. Please visit us.
Thank you, Mike. And next, I will turn over to Ed.
Hi, I'm Ed, coming here from New York. We offer dedicated Krios access on one of our 4 Krios instruments. We offer a grid preparation screening program using blood-free vitrification or normal plunge freezers and access to our glaciers. We loved seeing you at BPS, but if you didn't catch us at BPS, you can find out more at nccat.nyspc.org or our common website. One thing I would like to point out, we've been really heavily working on our merit badges, and the Legend on Screening merit badge is online. So Radhika probably can earn that if she hasn't already. So I'll turn it over to you, Christina.
Thank you, Ed. I'll stop sharing. So it is really a pleasure today to introduce our speaker, Dr. Radhika Malik. She's a research assistant professor at the Icahn School of Medicine at Mount Sinai, New York City. She has an extensive background in structural biology, starting during her PhD, where she used X-ray crystallography to study fundamental metabolic enzymes. But since then, she's also gained extensive experience in cryo-EM and has contributed her skills in structural biology and biochemistry to understand proteins and protein complexes involved in many processes, including neurological disorders and more recently, DNA repair and replication. She has contributed her expertise as a structural biologist in many collaborations and is a trusted collaborator not only in her role in science, but as a local cryo-EM champion at Mount Sinai, where she helps many other members of her department embark on cryo-EM projects. And her path on becoming that local champion included embedded training at NCCAT. And I just want to share my screen again to show you some photographic evidence of that training. So Radhika was part of a cohort at NCCAT during the winter of 2021, 2022. So it's been a little while now. But I think one of the unique opportunities Radhika had during her training was to actually get to look down inside the column of the microscope. It's not an event we look forward to having every day, but when we have those opportunities, they're a great part of training. So Radhika really has an in-depth knowledge of cryo-EM. And we're very excited now to have her back with us and find out how she's made use of her training and her other cryo-EM experience since to overcome some tricky sample issues on her research projects. So thank you, Radhika, for joining us, and I will turn over to you.
Thank you so much, Christina, for your kind introduction. It was really fun to be part of TP1. So let me share my screen now. The presentation here. So can everyone see the presentation?
Yes.
Okay, great. So I'm going to start by saying that today I'm going to talk about one of the most important polymerases in translesion DNA synthesis called DNA polymerase Zeta. And also I'm going to describe some of the cryo-EM methods that we use to decipher its high resolution structure. So for a long time, our lab has worked on eukaryotic replication. The replication fork is defined by a dozen of proteins, where these multi-subunit B family polymerases are the major players. We have DNA polymerase delta, which synthesizes the leading and the lagging strand. We have DNA polymerase epsilon, which synthesizes the leading strand, and polymerase alpha, which synthesizes the Okazaki fragment. So our lab has studied all these polymerases, but a major focus of the lab has been on these bifamily polymerases shown here in gray. So whenever a DNA encounters a lesion, these B family polymerases get exchanged by these Y family polymerases, which are usually these single subunit polymerases, except for DNA polymerase Zeta, which is a multi-subunit polymerase. So for a long time, we just had a negative stain model of this polymerase. However, with the resolution revolution in cryo-EM and recent advances in sample preparation methods, we were finally able to decipher its high resolution structure. And that's what I'm going to talk about today. So it may come as a surprise, but our cells experience DNA damage on a daily basis from various sources such as chemical carcinogens, ionizing radiation, and ultraviolet light, as well as several internal sources such as hydrolysis and oxidation. So our cells have various DNA repair pathways which they use to overcome these damages. However, there are some lesions which are left unrepaired, and that's where translesion DNA synthesis comes in. So translesion DNA synthesis is a central mechanism which happens in 2 steps, and it's basically a DNA damage tolerance pathway. So where once high fidelity polymerase, once it gets stalled by a DNA lesion, in the first step, Y family polymerase would come in and insert a base opposite the lesion. Once a base has been inserted, DNA polymerase Zeta would come in and put in a base opposite the mismatched termini. Once Polż has done its job, the high fidelity polymerase takes over, and then the normal DNA replication continues. So due to its importance in TLS and DNA repair, Polż has emerged as a target for cancers resistant to chemotherapy, especially those which are cisplatin-based therapies. And by deciphering the high resolution structure of this polymerase, we can then look at the various protein-protein interfaces and target those for drug development. So what is DNA polymerase Zeta? It's a crucial enzyme that allows our cells to battle DNA damage. It belongs to the family of high-fidelity polymerases called B family polymerases, and yet is able to do translesion DNA synthesis, wherein it acts as an extender and extends the DNA past lesions and mismatches. In terms of the composition of Polż, it's composed of the catalytic module Rev3, which has a polymerase domain and an inactive exonuclease domain. It has 2 cysteine-rich modules, cis-A and cis-B, at its C-terminus. In terms of the accessory subunits, it has Pol31 and Pol32, which it actually shares with the high-fidelity polymerase, DNA polymerase delta. Polż also has a third accessory subunit called Rev7, which is actually unique in all B family polymerases, and it actually belongs to the Horma family of proteins, which have other functions in the cell, such as acting as spindle assembly checkpoint proteins during chromosome segregation in the cell. So for a long time, it was thought that there is just one binding site of Rev7 in the RIR domain of Rev3. However, recently, a second binding site was discovered, and we were the first ones to characterize the dimeric Rev7 structure in the Polż holoenzyme. To start with, we started expressing and purifying the 4 subunit complex. However, we faced a lot of difficulty initially in characterizing the complex, primarily because of Rev3, which basically expresses very less and also tends to aggregate and pulls down the whole complex. However, by optimizing several conditions, we were finally able to get a stable and stoichiometric 4 subunit complex for a single particle analysis. So actually, we started off with the sample preparation for the apostate of Polż, and we used these holy carbon grids. At that time, what we were seeing was that the protein was not going into the holes, but it was sticking to the carbon. So what we did was we started using these lacy gold grids, which enable us, which enable the protein to go in the holes. And then we basically use these grids to collect data. We process that data, but lo and behold, we saw basically flat views of the molecule from our 2D classifications. So once we processed that, this gave us this anisotropic map from the 3D reconstruction. So these anisotropic maps are these smeared out maps, which basically misrepresent the true structure of your imaged complex. So what was happening? Basically, our particles were aligning to the air-water interface and giving us a non-uniform distribution of the angular projection views in vitreous eyes. So in order to tackle this problem, we started off by tilting the stage in the electron beam so that we could capture those missing views. However, when you do tilting, there are several considerations that you have to keep in mind. For example, there is always an increase in the beam-induced motion when you tilt, and therefore you need to focus on the grid support that you're using. And in this case, we use the gold grid support, which would minimize the beam-induced motion. Then there's always an increase in sample thickness once you tilt, and therefore you should consider either increasing the dose during your data collection, or you should optimize your grids further as well. Thirdly, there's also this defocus gradient, which happens across the micrograph once you tilt, and that you have to take that into account when you're processing. So we did a per particle CTF estimation, and we used GCTF for this purpose. So we took care of all these considerations, and we used a really clever app at New York Structural Biology developed by Angie. It's a tilting app in Legion, and we used that to collect our data set, and we were able to see the underrepresented as well as the missing views in our 2D classes. This resulted in a much more isotropic map. We also used CTF refinement, a per particle CTF refinement, to increase the resolution of our map. And then we used this FSC-based algorithm at the Salk Institute to check its sphericity. So recently, we also used tilting for another project in the lab, where we are again seeing these flat views. And then upon tilting to a high angle, we were able to see these different views of the complex. And upon processing, we now have a much better isotropic map where we can build very comfortably. So this was all about tilting and how we overcame the preferred orientation problem using tilting. So I'll go on to the ternary complex of Polζ now, where we were experiencing the same problem of preferred orientation and getting these anisotropic maps. So basically, in this case, we used Chameleon. So Chameleon is an innovative sample preparation method, which was actually called Spotted On and has been developed at the New York Structural Biology Center under the guidance of Bridget and Clint. And this novel sample preparation method actually overcomes a lot of problems, including a lot of problems with conventional freezing, such as protein aligning to the air-water interface, protein denaturation issues, as well as inconsistent freezing from one grid to the next. So what is Chameleon? So Chameleon is a blot-free technology where it uses these self-picking 9-layer grids. It uses a spraying technique to spray the sample on the grids, and that enables a specific control of ice thickness and also minimizes the variability from one grid to the next. In addition, it also uses the high-speed plunging. So basically, it doesn't allow enough time for your proteins to align to the air-water interface. And by doing that, it reduces the orientation bias. And it also reduces the protein denaturation effects that would happen during conventional freezing. So for the Polζ grids for the ternary complex, we optimized a spot to plunge time of around 130 milliseconds, and that worked brilliantly for us. So these are the results of the processing, which we also did using Topaz. That's a neural network-based particle picker that we used. And combining both the techniques, we were able to get all the side views and the other underrepresented views of the complex. And we were finally able to get a much higher resolution reconstruction of the complex to about 3.2 angstrom with a much better sphericity. So this is what the map of the ternary complex looks like. It has high resolution features at the center and low resolution features at the edges of the map. It was a very good map. And we were able to build a lot of de novo regions in this map quite comfortably because of the quality of the map. So let me describe the structure that we got. So this basically looks like a ring. The DNA is in red, and the only subunit that it binds to is the Ref3, which is in cyan. And then it has this iron sulfur cluster in brown at the C terminus, and it has these appendages which come out from the core of the Ref7 protein called the RIR domain, which wraps around these two Ref7 units. And to complete the ring, we have these Pol31 and Pol32 subunits. When we zoom into the different regions of the map, as I told you before, we saw great density for all the regions. This is the active site, this is the exonuclease site, and then this is the iron sulfur cluster site. So all these regions were brilliantly defined in our map. So if we zoom into the just the Ref7 structure and look at all the different domains, we find that the domains are in fact arranged in like a typical B family polymerase. However, there are some key differences. The palm domain, the fingers domain, and the NTD are actually much more elaborate, and we had to build a lot of de novo regions in these domains. The RIR domain is actually unique and doesn't exist in the other B family polymerases, and we had to build this entire domain de novo. Conversely, the CTD is present in all the B family polymerases. The duplex portion of the DNA is held by the thumb. And then if we zoom into the active site, we see that the T0 P0 base have these fingers closed in a closed conformation. So if we take this active site that I described just now and then compare it to a high fidelity polymerase, we see that the active site architecture between the two of them is quite similar. So this tells us that the features that impose high fidelity during the nucleotide incorporation step are similar to a high fidelity polymerase. However, when you look at the T1 site, this is where the lesion would come and sit in once a Y family polymerase inserts a base opposite it. The features around this region are actually quite different, and that is what I'm going to illustrate in the next slide. So here, Ref3 is in cyan, Pol3 is in gray, and the DNA is in red. So if you look at the linker, which connects the NTD to the palm, we see that it's quite closed in case of Pol3, whereas in case of Ref3, it has been pulled away by this palm loop, which is actually a unique element in the Ref3 catalytic subunit, thereby creating ample space for a lesion to sit and Ref3 to do its extension reaction. So now I'm going to highlight the second important subunit that we have from the structure, which is Ref7 dimer. So as I told you, it belongs to the horma family of proteins, which usually exist as dimers. They usually exist as the head-to-head dimers. By head, I mean that the C helix would form like a symmetric interface with the C helix of the other monomer. Whereas what we saw in our case was more of a head-to-tail dimer, where these C helices are actually quite far apart from each other. And both the Ref7s are interacting with the RIR domain with these specific RBM motifs, which are actually conserved across all the species of Ref3. So basically, the Ref7 dimer is the organizing center of the Polζ holoenzyme. So as you can see, the Ref7b here interacts exclusively with Pol31 and Pol32. Both Ref7s are interacting with the RIR domain of Ref3. And in fact, the Ref7 dimer has been concluded as as an adapter site for other TLS polymerases to bind during the TLS reactions. So since we also looked at the apostate of Polζ, we wanted to compare that to the ternary complex. And the main difference that we found there was that the fingers were actually open in case of the apostate, which is here in magenta, and the ternary is in blue. So it was open by around 15 degrees. And thereby it tells that Polζ's fidelity is augmented by the closing and opening of the fingers during the nucleotide incorporation step. So recently, we also determined the Cryo-EM structure of Polζ with a mismatch at the extension position. We used A instead of a G here. And the main difference that we found in comparison to the ternary complex was that the fingers were again open, were in an open state. And in an open state, the incoming nucleotide was still able to bind in the active site, thereby telling us a mechanism by which a Polζ would basically use to carry out and extend DNA mismatches and lesions. So in conclusion, we determined the Cryo-EM structures using novel sample preparation methods, and which unveiled the pentameric ring-like architecture of the holoenzyme. We identify structural elements such as the fingers domain and the entity palm linker, which would enable the polymerase to synthesize the DNA as well as perform translesion DNA synthesis. The holoenzyme had these two molecules of Ref7, which were arranged in a unique head-to-tail fashion, which has never been seen in the Horma family of proteins. And then the structure of the Polζ holoenzyme reveals critical protein-protein interfaces, which can now be targeted for drug development for the treatment of chemo-resistant tumors. So finally, I'd like to thank my advisor, Dr. Anil Agarwal, for all his support and guidance, Iban, Jakob, and Rinku for supporting and helping in the project at different stages. Misha for the Cryo-EM studies of the ternary complex, our collaborators in Texas for expressing the Polζ holoenzyme, and a special thanks to Bridget, Clint, Ed, and Yongzi for giving us a lot of good advice during the project. And also Zining for preparing those lacy gold crits that we used at the initial stages of our project. Alan Brown, he helped us with the model building and also provided help with the Balbus software that we used, as well as some de novo building that we did. And thank you all for your attention, and I'll be happy to answer any questions. Thank you.
Thank you, Radhika. That was
Thank you.
absolutely gorgeous. A lot of work packed into a short talk. So we've got some questions already. So there was a question about your tilted collection. What was the actual degree of tilt you used for your different data sets?
Okay, so for the ternary, for the apostate, we used 40-degree tilt. So we did try lower tilts, but that did not work. So we had to resort to a high tilt of around 40 degrees. And for the other projects also, it was high tilt for us, around 30 to 40 degrees.
Yeah, and actually you mentioned you were able to then get that structure with the mismatch. So did you have similar preferred orientation issues for that sample as well?
Oddly for that one, earlier we were not, so oddly for that one, we didn't have it actually. So, so that's the inconsistency in freezing with conventional grids, right? So we were using actually ultra AU foils here instead of the lacy gold grids. So I'm I think that could be a difference too, but yeah, we did not have preferred orientation for the mismatch complex, fortunately.
Yeah, that's intriguing.
Yeah.
And there's another question about tilting. For the higher angled tilts, what was the total dose? Was it much higher than for a non-tilted data set?
Yes, for the tilting, we use actually, so I don't mention that here, but yeah, we use around 80 electron per angstrom square. So we did increase it quite a bit, yeah.
All right. And then there's a question about which neural network software you use for the images. I'm guessing that might be a question about your topaz picking. If I'm mistaken, whoever asked that question, you can post a follow-up.
Yeah, it was topaz, yeah. Yeah, we use topaz for the picking.
Yeah, and actually with Topaz, did you train Topaz yourself, or did you use the pre-trained Topaz model when you did your
We
picking?
trained the topaz ourselves, yeah, with our particles first, and then use that again, that model again, yeah.
Okay, thanks. And actually, there's I like this thread of questions about tilting, so I'm gonna when I see those come in, I'm gonna stick there for a second because I think it's really
Sure.
useful. There's a question about what your ice thickness ranges were for tilted versus untilted data.
Okay, so for the Polž Zeta grids, the ice thickness, if I remember correctly, was of the order of 30 to 40 nanometers, 40 nanometers, 30 between the two, yeah, something like that. But there were, so we actually combined a lot of data sets here in tilting. We combined at least 4 data sets. So a couple of grids were not good. So we did have up till 50, I would say, 50 nanometers of ice thickness, yeah.
I have a question. It seems like you basically had two separate kinds of solutions to the preferred orientation problem. You tried tilting, and that worked for some things. You tried chameleon, which worked for other things. Do you recommend looking at all kinds of different ways to solve the preferred orientation problem, or do you see that you should try one first and then go to others?
I would say try everything. That would be my honest opinion, because tilting has its own drawbacks. Suppose you don't get a good grid, you are stuck with, you know, very thick ice, and tilting will not help you because you'll not be able to pick your particles, you'll not be able to see them. So that's tilting. Chameleon, you have to go to a very high concentration of your sample. I mean, I'm talking about proteins which are not very well expressed. So if you have to go to very high concentrations, then chameleon, there is a drawback in chameleon on that. And then graphene grids is another option which you could try. Actually, recently we have a project here which worked quite well on graphene grids, which I haven't talked about here, but you know, but actually for DNA protein complexes, detergent is something which has not worked for us for some reason. We have also tried detergents for our complexes.
Yeah, there's a lot of ways of solving it, and you know, basically you try everything.
But chameleon, if it works, is very brilliant. It's just a one-shot thing, and you get a beautiful structure.
Yeah, and actually you answered a couple questions that came in the meantime. There was a question about detergents. So I don't know if you had anything else you wanted to share about your experience trying them.
So we did try, if you remember, the V kit that I don't remember the company now. Do you remember that?
Yeah, I
Christina?
think it came from Jena Biosciences.
Yeah, you're right.
Yeah.
You're right. Yeah. So Jena Biosciences. So we have tried all the detergents in that kit, and none of them have worked for us as of now. So yeah, they have a bunch of detergents there. They have these, you know, the DDMs, NP40s, and whatnot.
All right. So I'm going to switch over. We've got a science question. Well, it's all science. We've got a biology question now. Zukai says, nice work. I'm surprised to see 2 Rev7 subunits in the complex. Do you have solution evidence for it?
So there has actually been an ITC evidence which was published at the same time we actually determined the structure. So so when we were building the model, we were not sure about this, another, you know, so if the second subunit. So let me go to the map first here. Maybe here is fine. So there's this 2 densities for at the bottom here. So we were quite sure that this was Rev7, but this density, since it was at the edge, it was slightly not as well defined, and we were not sure at that time what the second molecule, what this molecule was. Was it some of the disordered region in Rev3, or was it something else? So at that time, that paper came out, and that actually helped us. And we were looking at all these different de novo things, also used Balbus, and actually that predicted that it had a MAD fold, which is actually basically a hormofold of the protein. And so we knew that it had to be that, and that was confirmed by these experiments which were done by this lab here. Let me go back. So which I mentioned earlier. So yeah, the PNS paper which came out. So basically that confirmed through its ITC experiments that 2 molecules should bind in the human form of Polζ. So this is actually a yeast form. So still we had to make sure that it binds.
And someone related, actually, Luigi said, very nice talk. Thanks a lot. And asked how you expressed your protein complex.
So this was expressed in yeast by our collaborators in Texas.
All right, thank you. We've got a question about model building. So Andrew noticed that you used COOT for model building and asked if you used COOT to fit the DNA helix extending out of the enzyme as well as the protein, because they're struggling a bit with getting helical DNA fit into a map that they're working on, and was curious if you had any tricks for building the DNA, especially using COOT.
I would just use the restraints that you know, you would have for the DNA, and that's how we build the DNA in our lab. We use those restraints that are built in COOT, and those are quite nice actually, and they help in building the DNA quite accurately.
Yeah, and Andrew, if you wanted to follow up with more questions, I can promote you to a panelist and let you talk. Just let me know if you'd like me to do that. All right, I had a question for you as well, actually, Radhika. So your maps look so nice, and I think you sort of very nicely said how, you know, once you've got a really beautiful map, everything else becomes easy. So I'm wondering, does that mean that you had a very biochemically stable molecule to start with, or was there a lot of sort of triaging of particles during your processing? Like how much of how good your map looks came from the biochemistry versus the data processing?
So actually the sample was quite good. It was a uniform distribution of particles. We didn't have any heterogeneity as you see in many Cryo-EM data sets. So if I talk about this holoenzyme, then I mean, there are certain regions in Rep3 here which are missing, which are disordered, and we don't see any density for it. That is actually regions around this RIR domain. So it's around 300 amino acids which are missing in the Ref3 subunit. And we think that there are other factors in the TLS machinery which have to sort of bind for us to be able to see those regions of the RIR domain. Sorry, yeah. But in terms of processing, this was a pretty uniform data set, and there wasn't much heterogeneity that we had to sort out to get our ab initio models.
Great, thank you. There's a follow-up question about using Topaz. And the question is, I think, a very good question. So do not apologize for asking questions to our audience. Please send them in. Why did you use Topaz? Was it for denoising or picking better particles?
So basically for picking better particles, I would say, if you look at these classes here, and you see these side views here. So actually Topaz is really good at picking these side views in our data set. And if you read their article, it's actually, it has been known to pick these unusual shapes, which are non-spherical, which normal programs have difficulty in picking. So we did use template-based picking because we had a negative stain model initially. So we used those templates, but those templates actually were not that helpful in picking those these side views that we have here. So that Topaz really helped in that.
Thank you. So is Topaz a to-go picker for you, or do you reserve it for special cases? Or like, now looking forward to like other projects you've done since then, how often are you using Topaz versus not?
I would say all the time now, to be honest. Yeah, so and it has really helped in picking all the odd views, because all these DNA polymerases, they are anywhere near to spherical architecture, do not have anything similar to a spherical architecture. So yeah, Topaz really helps in that.
I have a kind of a basic biology question philosophically. If you have a mismatched DNA, is it smarter to try to fix it or to give up and say, if I've mismatched DNA, I know that something has gone wrong, and let me just kill the cell? It seems like if you try to fix a mismatch and you fix it the wrong way, you've introduced a mutation. Or are these things happening so often that you have to try to fix problems?
So they are happening to some extent, especially if you have these chemoresistant tumors. So whenever you have radiation impinged on you, it generates all these mutations, right? So if we and Polζ what it also does is tries to fix those, right? So by bypassing them and creating these chemo-resistant tumors. So that's one thought where if you mutate Polζ in a way that it becomes inactive, then these chemo-resistant tumors would basically reduce.
Yeah.
Right. So that's what the thought is, and that's the importance of this protein actually, in the and that as the importance of the pathway itself. I'm not just saying this protein, there are other proteins also which it interacts with, and those all have very critical roles as well in doing all this. So yeah, so that's why we want to study all these proteins and just develops drugs to basically inhibit this pathway, and that would help with cancer chemo resistance. I don't know whether I answered their question properly, but
yeah, I see. Yes.
okay.
You want to design a drug to inactivate it.
Yes, yes. Yeah. True. And this is a last resort mechanisms. There are other better DNA repair mechanisms in normal cells, right? So which help in repairing our cells on a daily basis. But in extreme cases, these repair pathways become predominant, and then they create havoc, and how to stop that is the question.
All right, we have a question here from Miriam about combining data sets. And the question is, did you have any issues with duplicate particles or issues with anisotropy when you're combining multiple data sets?
So we do remove duplicate particles in the end. So yes, we do have issues with duplicate particles, and we do end up removing them in our processing pipeline. And what was the second question, Christina?
And whether that leads to anisotropy.
Yeah, so we do remove them. So yeah, so there is no reason for that contributing to anisotropy.
And what software are you using to do that processing and make sure you're removing duplicate particles?
So we are using actually CryoSpark now to remove the duplicate particles. Yeah, and processing in general, we have done both, I mean, in CryoSpark initially, and then we also do rely on as well.
All right. I believe that answers that question. If there are any more questions people have, go ahead and send them in. And if we don't have more questions, we'll stall for a bit while people have a second to type, because often typing takes an extra second.
Sure.
But yeah, if
Should
there,
I stop my share here?
Yeah, go ahead.
Okay.
Yeah. If we don't have other questions, thank you, Radhika. I think you gave us all a lot of ideas. It's really great to see you were able to overcome those challenges and find multiple ways to try to do it. And so thank you for sharing that with us. And we hope all of you out there doing Cryo-EM also have as good luck overcoming your challenges. And here at the National Centers, we're here to help you. So let us know how we can do that. And we will see you all here again next month, hopefully. Thank you.